The year is 1970, and a 53-year-old woman is fighting for her life in a hospital. She's been battling chronic myelogenous leukemia for 4 long years, trying treatments like busulfan and pipobromin, but the cancer kept getting worse. By November, her condition had become critical. The cancer had spread throughout her body, forming tumor masses in almost every tissue. Her chest was filling with fluid, making it hard to breathe. 9 days before she died, doctors took a sample of that fluid from around her lungs. Those doctors had no idea they were collecting cells that would become one of the most important research tools in medical history. When researchers Lozio and Lozio published their findings about these cells in 1975, they described something extraordinary. These weren't just any cancer cells. They carried the Philadelphia chromosome, a genetic marker that scientists were just beginning to understand. More importantly, these cells did something that normal human cells just don't do. They kept growing and growing and growing. These K562 cells have become what scientists call an immortalized cell line, and they've been used in thousands of research studies. They've helped develop treatments worth billions of dollars, including drugs like Gleevec that have transformed cancer care. In today's Masterclass episode, we're diving deep into everything you need to know about K562 cells. We'll explore the science behind what makes these cells so special, including that Philadelphia chromosome and the BCR-ABL fusion that drives their immortality. You'll discover how these cells have been used to develop some of the most important cancer treatments of the last 50 years, and why they're still crucial for cutting-edge research happening right now. Joining me today is Dr. Tom Karagiannis, who has been working with K562 cells for almost 3 decades now, and can give us the inside story on both their incredible potential and their limitations. We'll get the real scientist perspective on what it's like to work with these cells, how they're being used in current research, and what the future holds. Whether you're a student trying to understand cell biology, a researcher working with these cells, or just someone curious about how medical breakthroughs actually happen, this episode will give you everything you need to know about one of science's most important tools. Okay, amazing. Dr. Karagiannis, welcome to the show.
Thanks very much, Mike. Thanks for having me.
It's 7 a.m. here in Toronto and 9 p.m. Over there in Melbourne, so it's definitely going to be an interesting show.
Interesting.
Let's talk about K562 cells, and let's start just by the elevator pitch. What are the K562 cells and why they're important?
Yeah, so K562 cells are essentially a chronic myeloid leukemic cell line. They're called erythroleukemic because they're derived from a red blood cell progenitor, and they were developed by a team at the University of Tennessee, and they're one of the very early continuously growing cancer cell lines, if you like, and they have a very specific chromosome known as the Philadelphia chromosome, which we'll get into in this episode.
Mm-hmm.
So they're a chronic myeloid leukemic cell line for listeners out there.
Mm-hmm. Nice. And so you mentioned to me before the show that you've been working with these cells since 1996, which is 2 years before I was even born.
Excellent.
So what drew you to them initially? What experiments were you running back in 1996 with K562 cells?
Yeah, so Mike, in 1996, I was a summer vacation student at the Peter MacCallum Cancer Centre here in Melbourne. So it was a competitive process straight under undergraduate, and there was I was doing an internship, a summer vacation internship at Peter Mac. And my supervisor said that for my honours project, I'll be using K562 cells as a model for targeting transferrin receptors in order to target different drugs to using the transferrin receptors. And because they overexpress transferrin receptors, they're a good model system. And my supervisor, drew my attention to these cells. Immediately, of course, I was young at that time as well. I was in my 20s. Straight out of university. And when I read about them having a Philadelphia chromosome, and linking that with Rocky, who's also from Philadelphia, I was very, very
Mhm.
excited. Rocky's the boxer, you're
Oh
probably too
yeah,
young, but yeah, right,
yeah.
yeah. So
Mhm.
Philadelphia drew my attention, and I thought, oh my God, I love these cells, and I love the Philadelphia chromosome. So
Mhm.
that's
Nice.
yeah,
Yeah.
yeah,
So okay.
excellent. So that was
Yeah. Back then, 1996. So these cells had already been around for like 2 decades, right? Or like
correct. No,
maybe even yeah. So um, I mean, like, how did it feel back then to work with these cells? Was this like the new thing, that everyone wanted to work with, or like they were skeptical about this? Like, what was the general perspective
no,
of this?
no. So I was, I could say, lucky in a way. When I first started in 1996 in imatinib, and again, we're going to talk about this later, but in imatinib mesylate, right, Gleevec, as people would probably know it started becoming not available, but there was a lot of talk about these tyrosine kinase inhibitors. So it was a very,
Right.
very exciting time, especially a lot of cancer researchers at the time. They were saying things like, that's the end of cancer research. These tyrosine kinase inhibitors, like targeting this Philadelphia chromosome, they're the next big thing. That's it. It's all over. Yeah. Because as we'll see in a minute, when we talk about personalized therapies, but yeah, there was a lot of excitement at the time about using K562 cells because of the Philadelphia chromosome and because of Gleevec as well.
Mm-hmm.
So
Right.
despite my rocky story, which is interesting, there was also a very good scientific
Mm-hmm.
basis for using these cells at the time. And again, Mike, I want to tell people that are listening and yourself, at that time, even though it was 2 decades that they were available, cell culture was very rudimentary. It's not like we had glass pipettes, to give you an
Yeah.
example, right? We were using glass pipettes. It was a very different ball game to what it is now. It
Uh-huh.
was special to work with human mammalian cell culture, we used to call it. Yeah, it was very special.
Right.
Yeah.
What is the actual origin story? Like, where do these cells even come from?
These cells come from a team. So they're published by Lozio and Lozio, right? And given the surname, I assumed they were a couple or something of that sort, right? But all my
Mm.
research is showing me that it's very no one knows if they were a couple or if they were like siblings or what they were, right? So no one
Mhm.
understands that. But anyway, it was a team called Lozio and Lozio that
Mhm.
first published this cell line. Um, and I think it was in 1975 that they actually did the
Mhm.
publication, but they were working on them a bit earlier. And they were at the University of Tennessee, and they had a patient who went into blast crisis. So blast crisis is the very end stage of chronic myeloid leukemia. That's the stage where you get really rapid proliferation of the cells and you get a pleural effusion. So all that means is that there's a lot of fluid around the lungs. So they took some of that fluid out of the lungs and then they grew those in, I think it was Eagles medium back then.
Mhm.
And they found that these things grow continuously. So
Mhm.
that's the story. Yeah.
Right. So,
Mm.
and well, like when someone says like they established the cell line, I mean, like what does that even mean? Because like if you just tell me like here's the patient, go establish a cell line. I mean, all I can picture is just like I need to draw some blood and put it in some media, hoping it grows. There's something like that. Like, what
No.
is the
Yeah,
actual procedure?
no, it doesn't work that way because what would happen, and this is why a lot of attempts failed earlier. What happens is these cells they might divide. Like example, if you take white blood cells out of a person, right? So if you take normal blood, peripheral blood, you will grow for 24 hours, maybe 48 hours, and then it's going to die, right? So they usually just die in media. And a lot of the cells that they used to have before, they required a feeder layer, a feeder layer. So what that means is they will grow them on a Petri dish on a layer of cells that had been irradiated with 30 gray, right? So a big dose of gamma radiation. And what that does is that kills the cells. But by killing the cells, they release all these nutrients, and
Uh-huh.
those nutrients help your cells grow. Get it? So it was a feeder
Uh-huh.
layer. Yeah. It was
Uh-huh.
so that's what I mean by a continuous cell layer that did not require a feeder, right? So they take them from the lungs and they put them in a special medium which has got all the nutrients, like all the amino acids and all the iron and everything that they need to grow. And you also use inactivated fetal bovine serum, right? So you take bovine serum, you inactivate it with heat, so you kill off any antigens or anything like that might kill your cell. And then you use that with specialized media, like a red liquid, which has got all the amino acids and all the growth factors that you need. And then you put your cells in there, and that medium can then sustain it at 37 degrees. So you put those in an incubator at 37 degrees with oxygen. These days, incubators, you'll see they come with 5% CO2 built in. But back in the old days, when we used this cell culture, we used to have to put in nitrogen oxide into the flask, seal it, and then that's how we could put in the CO2 required, right? With actually a gas cylinder, a CO2 cylinder. And that's why it was very difficult back then. It wasn't as easy as it is now to do cell culture. It was the much harder thing. And as I said, a lot of the cells required a feeder layer. So a layer of cells that were irradiated or, with a very, very heavy dose in order for them to die and then to release nutrients to make the other cells grow. And these cells were the first leukemic cells that didn't require a feeder layer, that could grow on their own. So that's why they're interesting.
Okay. Yeah. So I'm thinking like I'm wondering, because I assume like there were other attempts at creating CML cell lines, but we don't hear about them. And K562 made it all the way to like 2025. So I'm just like wondering what made the researchers so successful at this attempt. Was it just like they used some like new molecular biology trick, or they just got lucky with timing, or?
I think it's a combination. So it's a combination. What happens is at that time, even the media, I'm talking about media, like Eagles media, at that time, these media were getting better and better. People were understanding, because what preceded these cells was the HeLa cells. Of course, they're the very first cell line, the HeLa cells. Everyone knows the HeLa cells from Henrietta Lacks. Everyone knows the story. And so they're the first ones. And then as they were getting cultured, even cell culture techniques were improving more and more. And then the growth media and grow the cells in was improving as well. So there was a thing called Eagles modified media, which was a special liquid with all the amino acids and all the nutrients, and the use of fetal bovine serum in order to provide more proteins and growth factors for the cells. So then it was a good combination of many things coming together. And one more thing for perspective for people to know, and I'm reading a very interesting book right now. It's called Cancer Virus. And it's all about the Epstein-Barr virus and Burkitt's lymphoma. So the very first cancer cells were these Burkitt's lymphoma cells, and they were virus activated from the Epstein-Barr virus, right? So they could grow cells, cancer cells, that were transformed with the virus, but they couldn't grow cancer cells that weren't transformed with the virus. And K562 cells were at the right time when all the media was getting better and all the cell culture techniques were getting better, and then they could grow these without a viral infection, like the Epstein-Barr virus. But the book I'm reading is called Cancer Virus, and it's all about Burkitt's lymphoma and the history about how they identified that cancer virus. It's very interesting.
Nice.
People are interested.
Well, I mean, if you have it, maybe we can show it to camera. If it's
I'll try to show it to the camera here. It's called,
okay.
Yeah, cancer virus.
Nice.
Yeah.
Amazing. Okay, so let's talk about the characteristics of K562 cells. I have some items on my notes, and let's start with the Philadelphia chromosome. What is the Philadelphia chromosome, and why is K562 having this so important for us?
Yeah, so the Philadelphia chromosome, so even before K562 cells, people had recognized the Philadelphia chromosome. And the reason it's called Philadelphia is because the two scientists that first identified it, I think it was about maybe 15 years earlier or something like that, they were from Philadelphia. And that's why they named it the Philadelphia chromosome. They worked at, And what it is, it's a translocation. So it's part of chromosome 9, and part of chromosome 22 that have swapped over, they've translocated. And what we get is some material on chromosome 22 from chromosome 9, which forms this which makes the Philadelphia chromosome. Okay, so it's this the truncated chromosome 22 is the actual Philadelphia chromosome. And what that does is it brings the BCR gene, right? Like the breakpoint cluster region gene, together with the Abel protein tyrosine kinase. Okay, so it brings those two together, and what you get is a constitutively activated tyrosine kinase, right? Which then drives proliferation, right? So this new protein that's made continuously drives proliferation. The cells just keep dividing all the time, despite signals. They don't need any signal from the outside. Yeah.
Right. Which I assume has a role in cancer.
Of course. So cancer is is all about proliferation, right? So the definition of cancer is a cell that can divide and form, progeny without receiving any signals from the outside. It's got its own, own ability to do it without driving forces. So it's bypassed all the regulatory factors that you usually have, right? Yeah.
So when you have the Philadelphia chromosome, so both of your chromosome 9 and 22 are messed up. It's not like you have a, so because I just wanted to clarify, like it's not like you have chromosome 9 and chromosome 22 and then another chromosome. It's just like they're both translocated.
They're both translocated. They're messed up.
Right.
Yeah.
So, and you mentioned like K562 cells have this Philadelphia chromosome, which makes them so special as a model to do research on the Philadelphia chromosome. But why do we even care about the Philadelphia chromosome? We touched on the cancer thing.
Yeah. So as I mentioned briefly earlier, so the Philadelphia chromosome is has given us the opportunity for the first targeted therapy in cancer, right? So when we talk about personalized therapy, this was the actual first personalized therapy, and it was Gleevec, imatinib mesylate, right? It's a tyrosine kinase inhibitor which specifically targets the BCR-ABL fusion protein that's formed when when the Philadelphia chromosome is formed through the translocation process. That's the important part. Yeah.
Nice. And so the K562 cells are able to proliferate continuously for 3.5 years while retaining their Philadelphia chromosome. So why is this? Well, I mean, I guess like we talked about this, but I was just gonna ask, like, why is it the Philadelphia chromosome positivity is so crucial? But I think we talked about this.
No, that's fine. And again, I'll reiterate, it allows for targeted cancer therapies, personalized therapy, which is the big thing these days, right? And if you think about it, it's taken 30 years since imatinib was discovered in order to come up with new tyrosine kinase inhibitors. And these days, today, the research is focused on these, and a lot of the clinical trials in cancer are related to these tyrosine kinase inhibitors for different types of cancers.
So this Gleevec
opened the way. Yeah.
Right. So this Gleevec targets the Philadelphia chromosome?
It targets specifically the BCR-ABL protein that is made because of the Philadelphia chromosome, the new protein. And that's why it's a management for cancer rather than a treatment. So it doesn't cure the cancer, it manages the cancer. So I know people that have been on Gleevec for 20 years, or over 20 years even. Yeah. And it just manages. They can't come off it. Yeah.
And when we say cancer, in this case, we specifically mean like CML?
CML. Yeah. Chronic myeloid leukemia, specifically. Yeah.
Right. Nice. So next item that I have on the list of characteristics is K562 cells are pseudo triploid. Can you tell us what that means?
Yeah. So as we know, normal cells have like 23 chromosomes each. pairs. With K562 cells, they're nearly triploid. They're not exactly triploid. They're almost triploid.
Uh-huh.
So when you look at the karyotype, and we've done a lot of karyotype studies in our paper, you
Uh-huh.
get like maybe 4 copies of one chromosome, 3 copies of another chromosome, 2 copies of another chromosome. So altogether, it's almost a triploid, right? But it's all messed up. Some chromosomes you get 4 copies, some you get 3, some you get 2. It's a mess.
Uh-huh.
And you also get the Philadelphia chromosome. So that's what we mean by pseudo-triploid, almost triploid. Usually they
Right.
have, I think it's between 70 chromosomes, let's say. Approximately 70 chromosomes.
Wow.
Yeah.
And so the other interesting thing about K562 is that you can push them to differentiate into many different specialized cells. And can you briefly talk about this as well?
Yeah.
How do you actually do it in the lab?
In the lab, yeah. So I've seen that by accident, to be honest. I haven't tried to differentiate them in the lab, but because I've used them for so long over many years, I've treated them with different things, HDAC inhibitors and different drugs and radiation and all sorts of things. So I've seen the cultures like differentiate unknowingly that I'm differentiating them because I've seen massive megakaryocytes and I've seen different type of morphologies inside the cultures. And but you can do that specifically. So if you use EPA, for example, you're going to get an erythroid cell. If you use you can use different factors to push them into the lineage that you want. Yeah.
Right.
Yeah.
So
Yeah.
here's what I don't understand. I mean, if you want to study CML, K562, amazing choice, no doubt. But like people, so for example, they want to study megakaryocytes and they treat K562 with PMA to get this like megakaryocyte like K562, and they use that to do their research. But I mean, we talked about the abnormalities of K562. They have this like the Philadelphia chromosome, the pseudo-triploid. They're just so messed up in many different ways, which makes me wonder, like, why do you even want to use them to like study normal human cells?
Yeah, I get that. I get that. And when I was younger as well, I used to think, "What the heck's going on here? Why are we doing this?
Yeah.
We could do better, right?" Yeah, I get it. But the thing is convenience as well, right? K562 cells, you can get huge numbers of cells. Now, if you differentiate them using an easy factor in the lab, right? So you use some kind of some mirror state or some easy compound that you can buy, you're going to get huge cell numbers. That gives you the advantage of being able to do many different types of experiments. Like, you can do sequencing, you can do PCR, you can do whatever you want. So the idea is it's convenience. I know it sounds silly that you'd go to that length and then to use a cell type that's probably not going to be relevant, but it's relevant enough, and the convenience gives you that advantage that you think, okay, I want to do it this way because at least I'm going to get the cell numbers. To get primary megakaryocytes to grow, you won't get the numbers. It'll be very difficult to get those numbers. Yeah, we won't be able to do the experiments. But look, I used to think like you as well, especially at a younger age. I used to think, what the heck? Why are we doing it this way? This is crap. We could do much better. Yeah.
Um, the other thing that I have is that K562 cells massively overexpress transferrin receptors. Can you
Yes.
unpack this for us?
So
What is a
let me.
transferrin receptor even?
Yeah. So transferrin receptor is expressed on the cell surface on the membrane of K562 cells, and it undergoes endocytosis, right? And this is my honors and my PhD, so I know this inside out. I studied this. This is what I did, right? So K562 cells express 150,000 of these receptors, okay, on the cell surface. What they do is they get internalized. So what we've got is we've got iron, in the plasma. They bind to iron. 2 iron molecules bind to transferrin. So it gets internalized through an endosome. It then releases the iron molecules, right? So it's an iron transporter. It releases the iron molecules through a reduction process, and then it gets recycled back to the cell surface. So it's a cycle. It's a transferrin receptor mediated endocytosis cycle. Okay? And the idea is that it gives the cells iron, and cancer cells need a lot of iron in order to divide. So that's why they've got so high levels of transferrin,
Right.
because they need to
How
metabolize iron. Yeah. Sorry?
how does it relate to cancer?
Yeah. So cancer cells are rapidly dividing. Okay, so they're dividing all the time. And one of the early experiments that I had to do with K562 cells was a doubling time, right? And K562 cells double every day. So every 20 hours they'll double. So that means they need a lot of nutrients, right? And a lot of iron. So
I
they
see.
express a lot of iron in order to be able to divide and form new copies of themselves. Yeah. Yeah.
Yeah, I think we're painting a great picture explaining why like these cancers happen. We have the Philadelphia chromosome, which gives us that protein, and we also have just like this massive supply of nutrients into the cell. So everything like makes sense now. And so we talked about the Philadelphia chromosomes that can be used as a biomarker. What about this transferrin receptors? Anything about them can be used as a targeted therapy?
Yes. So we can, with one, and I have to be honest, with one caveat, right? So there's one caveat. Transferrin receptors are also, lots of them are expressed in the liver, right? And the liver is a huge organ. So it's very good for cell culture model systems for studying. But when you come to doing, say, mouse experiments or doing experiments in humans, what you'll find is all of it will go to the liver, because the liver acts like a sink for iron. That's why, when you're low in iron, you eat liver, because it accumulates all the iron, right? So there's a lot of transferrin receptors. And when you do in vivo models, unfortunately, the transferrin receptor as a target falls apart because of that. Yeah.
Right. So we're better off just sticking with the Philadelphia chromosome as a biomarker.
As a biomarker, but also, also because transferrin is very cheap, as we said before, right? Sometimes convenience. It's a very convenient model system. Let's say you're making a new drug, the type is a transferrin receptor, or type is, let's say you're making a drug that is modular and you can type it anything you want. A lot of the problems will be chemical problems at the start. So this system gives you a very cheap system in order to operate, right? For the transferrin system. Transferrin, you can buy it. It's very easy to buy, very easy to get. It's very cheap. And you've got this system with a lot of transferrin receptors. So it's a good model system to start with, to start your experiments. And then if your chemistry works, you can then change the chemistry for another system, like another antibody-based system. Yeah.
Nice. Let's talk about some of the technical considerations when it comes to K562. For researchers working with K562 cells, what are the key technical considerations for maintaining all these characteristics that we talked about?
Yeah, as I said, one of the beauties of K562 cells is the ability for them to grow. Okay, so they are, we say they grow like weeds. They're like weeds.
Yeah.
It is very hard to stuff up a K562 cell culture. Even the one where you overgrow it, you can just take a little drop, put it in fresh media, because they're a suspension culture, right? So they don't stick to the plate or anything like that. So even if you're overgrown, you can take a little drop, put it in fresh media, and in 3-4 days you will have fresh cells growing again. So they're very easy to maintain in cell culture. One of the easiest cell lines to maintain. Yeah.
And
The only thing you don't want to do, Mike, is to use overgrown cells in an experiment, right? Because what happens then is all your gene expression profiles, your transferrin receptors, everything changes because they're overgrown, and you get like a mixed population. You want to use them when they're in exponential growth phase, right? So when you've got about up to 1 million cells per mil, that's when you should use them for experiments. Anything above 1 million per mil, it starts to overgrow. And what you should do then is culture them. Just take a little drop, put them in fresh media, wait a couple of days, and you'll get fresh cells again. Yeah.
So what is like a safe number of passage with these cells? Like, is there like a limit, like after 10 passages, you don't want to use them, or like?
No, Mike, even as you said, 3.5 years was the initial. In
Uh,
our paper, we grew them for 500 days. So we did an experiment that went for almost a year and a
mhm,
half. As I said, they do, as we'll see, they do, there's really some genetic drift, but they are a continuous cell line. They've been going forever. Yeah.
mhm,
Mhm.
nice.
You're not limited by
Um,
number of passages with these ones.
mhm.
Mhm.
Um, I'm just wondering, like, with everything we know about K562 cells now, can we just like easily, um, like get, uh, I don't know, K562 like cells from CML patients now? Like, do we have like the protocol? Like, we don't need, because like back then, of course, you talked about the technical limitations, but given everything we know about K562 cells now, can we just like replicate the success with like every patient?
Not every patient, but we can, yes. It would be much
Mhm,
more successful these days at doing
mhm,
that. And there's patient-derived cell lines these days that people use readily in the laboratories. There are patient-derived
mhm.
cell lines which they can actually grow and they can inject into mice and get tumors. And and I'm not sure if you'd be able to replicate the K562, the luck that they had then, that you would get
Right.
one that will proliferate indefinitely, but you will get some divisions for sure. Yeah. Yeah.
Because I'm just wondering, like, if we can just easily do that, um, can we like make personalized K562 cells for each patient? But also, like, is it even worth it? Like, you get like some mutations, but I mean, because like when we talk about doing it practically and like in massive numbers, like, is it even worth it?
The thing there, Mike, is the ethics, right? So you would need ethics
Mm,
for every single patient, for every
mm,
single. That's what the limitation would
mm,
be. Again,
right.
we've got a cell line that you can grow straight away and have no problems. To then go to the effort to get ethics for every single patient, and then even then you're going to get mixed results because it's not going to be a perfect isolation every time. There will be difficulties. Yeah. Whereas here we have an established cell line, it's well characterized, much easier for everybody. Yeah.
Mhm.
Mhm.
And so when you look back on your career over the decades, how have the primary research applications of K562 cells evolved?
So a lot of the early research, because it depends on the era. So science is cyclical, right? So
Mhm,
in my era, it was all about targeted therapies, right? So we wanted to make like conjugates, immunoconjugates that will target and kill cancer cells, right? So an antibody with a drug or radiation that will kill cancer cells, right? It was all about cytotoxicity and things like that. In the next phase, it was all about the kinase inhibitors like Gleevec
uh-huh,
and all different types, right? So there was massive screening of tyrosine kinase inhibitors. Then came the sequencing era, right? Next generation sequencing, where you can then do the microarray analysis and next generation sequencing. So it's all about transcriptome and transcriptional analysis. And then, I mean, the most recent is the CAR T, development of CAR T cells. And
right.
again, in this case, K562 cells were used as a workhorse because you can grow them to huge numbers. They'll use as a workhorse for the development of CAR T.
Hmm. Is it like everything we find with K562, is it just specific to CML cancer, or can it be like applied to all the other types of cancers?
You could loosely apply it to leukemia, because
Uh-huh.
they're rapidly growing proliferative cells. There are some depending on if you want to do AML, because AML is the big killer, right? Acute myeloid
Hmm.
leukemia. That's the one where it's very, very bad. And there are good cell lines. The HL6 is a good cell line for that. So if you wanted to study AML, you'd probably use that cell line. And for CML, this is the best cell line for the CML, but you can. I mean, it is a model for other tumors as well because it does have a lot of the characteristics.
Great. I think we have laid a fantastic groundwork to talk about your paper. You published this paper, Characterization of K562 cells, uncovering novel chromosomes, assessing transferrin receptor expression, and probing pharmacological therapies. And let's talk about that. So first of all, when you say novel chromosomes discovered in prolonged K562 cultures, what do you mean by that? Can you unpack that
Yeah.
for us?
Okay. So our cell line, when we first got it in the lab, as I said, I got it in 1996 from another lab. And then I used it for many, many, many years, right? So I thought, let's do a karyotype and see what happens. So we did some karyotype analysis, and that's where we identified a novel chromosome that had never been described before. And so that happens. So when you have a cell line in culture for 20, 30 years, that can naturally happen when you get chromosomal translocations and aberrations, genetic drift over time. But the interesting thing is we found a chromosome that hadn't been characterized before.
And so for how long have you grown these cells to see the novel chromosome?
So again, we didn't do a time course. This was done by accident. It wasn't done with a specific goal in mind that one day we're going to look at the karyotype and see what happens. Because then we would have had it before and after. That would have been interesting, right? But
Yeah.
I think so. I got him from someone in 1996, and then the actual experiment we did here was probably 2016. So yeah, 20, 30 years later. Yeah.
And I'm asking this as someone who has no idea about karyotyping, but I'm very interested. How do you actually do karyotyping?
So karyotyping, that's a good question because we don't do it. It's a specialized lab. It is art. It's an art form. So yeah. So they remove the chromosomes. They have special techniques. They remove, then they take pictures of the chromosomes, X-rays. There's another experiment here where they've done immunofluorescence of the different chromosomes. It's called FISH. So, and as I said, these are very specialized labs, laboratories. And they're more like artists than scientists. I mean, it's a craft. It's a craft.
Yeah, I can tell by just looking at the pictures that they do. So one of these novel chromosomes is their chromosome 6, the derivative form of chromosome 6, with chromosome 16 material insertion. Can you tell us about that?
Yes.
What does that actually
okay.
mean?
No. It means it's a novel chromosome that's no one has ever seen before. Functionally, we didn't study it, so we don't know functionally what that actually means for the cell. But yeah, that's a total different study as well to look at the functionality of what that actually means. Yeah, but literally, it means what you've said is it's a new chromosome that's come together as a part of the culture process. Yeah.
Yeah, and the other one that I have on my notes is you had a derivative chromosome 5 with translocation with chromosome 4, and this one was actually very common in your samples. 71% of cells had this chromosome, right?
Yeah, 71% of cells. So yeah, so the implication for this is when people do, especially next generation sequencing or when they're looking for very specific things, then it'd be, how sequencing works by aligning it back to the genome, right? As you can see with the K562 cells, the genome's all messed up. So it would be very difficult to draw conclusions from NGS experiments, for example, unless you had the annotation. What you need is an annotation for K562 cells, which is specific, in order to go back and then trace exactly what's going on. Yeah.
It just came to my mind. like when something like translocation between chromosome 4 and 5 happens, or like 9 and 22, like why is it like these two specific chromosome? Is it just like pure luck, or like is it because they are in a specific location in cells that makes this like enables this process, or like do we know something about like why these two specific chromosomes?
Mike, we do. So, and this comes from radiation studies. So when you radiate cells with gamma radiation, for example, you get a lot of translocations, and we don't know exactly why it happens, but you get similar types of translocations. So there is some reason as to why there is these that are coming together and not something else, right? So you're right. There are patterns. There are patterns that occur, and we've seen this with radiation, because radiation causes chromosomal translocations. It's one of the things that it does. And there are patterns,
Right.
but again, very interesting to see exactly why that pattern's happening. Yeah.
Yeah, and you mentioned radiation. I just instantly thought of skin cancer and UV. Maybe like, is this related? Like, maybe like UV causes some specific translocation in skin cells and.
Correct. It does cause translocations and pyrimidine dimers. And yeah, absolutely.
Interesting.
Yeah.
Let's go back to the
You
questions.
you'll need
Sh-
a 1970s, a little bit older than me even, radiation biologist. So one of the guys who was studying radiation biology back in the day, because they were all about chromosomal translocations and
Mhm.
exactly what's going on. They'll know much more than me for that about that. Yeah.
Mhm.
Mhm.
And well, I mean, I suppose it's like one of the best biomarkers for cancer because it's so specific, or.
It is. I mean, translocated chromosomes, as you know, are very serious, right? So a lot of
Mhm.
them are deleterious. They'll kill the cell. And yeah, absolutely. Yeah.
Mhm. So the other thing that you mentioned on your paper is about transferrin receptor expression and regulation. So your paper confirmed high transferrin receptor expression persists even in doxorubicin-resistant
Yep.
K562 cells. Why is it important clinically?
Yeah. So we did that experiment. So we did that experiment with doxorubicin. So basically what we did is we did escalating doses of doxorubicin. And for people who don't know, doxorubicin is a frontline anti-cancer therapy. So it's a cytotoxic agent. It's a frontline cancer therapeutic that's used in cancer patients. It's called an anthracycline, and it kills cancer cells, right? But what happens is inevitably cancer cells will get resistant to doxorubicin.
Uh-huh.
So what we did is we took K562 cells and gave them small doses of doxorubicin, so we don't kill them, right? We keep them alive, but they start to accumulate resistance. And we did the experiment over 500 days, so 1.5 years. And by the end, we had a doxorubicin-resistant cell line that we developed in our lab, right? And the normal K562 cells. Then we compared transferrin receptor expression to see if that changed, and it didn't, right? So that means the cells now are resistant to cancer therapy, but they still have the metabolic capacity to divide. So they can still drive carcinogenesis. That's the implication, right? The cancer cell will still survive. You'll still get all the nutrients it needs, but chemotherapy won't affect it at all. Yeah, yeah.
So bad news for
Bad news, but yeah, bad
yeah,
news
for
basically.
cancer research.
Yeah.
Um, you also found that epigenetic modifications regulate transferrin receptor expression. Could you explain what specific histone modifications you observed?
Yeah. So we did what's known as a chromatin immunoprecipitation assay, and we looked at the promoter region of the genes to see if it's acetylation, because acetylation drives. There are different markers, right? Epigenetic markers, acetylation and methylation on lysine free. I won't go into epigenetics. That's a whole different field for people. And we can talk about that another time. But yeah, everyone will get confused. But basically, we looked at permissive markers, epigenetic markers, that allow transcription to occur, right? So acetylation is an example. Methylation on lysine 3 is an example. So what we found is that these permissive markers at the promoter region of the transferrin receptor allow the transferrin receptor to be expressed.
Mm-hmm.
Mm-hmm.
So when you say you did this with genes, what genes specifically did you look into?
So again, the ones that we showed here is the transferrin one, and we looked at histone acetylation, and we looked at histone methylation as well, right? So permissive markers at the promoter region or around the promoter of the transferrin gene. And we did that in K562 and in normal cells. And we found that in K562 cells, these permissive genes allow the expression of the receptor, these epigenetic marks. Yep.
Right. You found that K562 cells overexpress class 1 and class 4 HDACs compared to normal cells. What does that mean in terms of like clinical implications, and what does even HDAC mean in the first place?
Correct. So HDAC, they're histone deacetylase inhibitors. So I just talked about acetylation of histones. So acetylation of histones allows histones, this is very broad what I'm saying, allows histones to transcribe. So active transcription. Histone deacetylases remove the acetyl groups, right? So they block the transcription. So the inhibitors of deacetylase enzymes then cause histone deacetylation. Okay. So this is very confusing. It's hardcore epigenetics, but the bottom line is by using these HDAC inhibitors, we can kill the cancer cells, right? And HDAC inhibitors like trichostatin A have been used experimentally. And there's one, it's called SAHA or Vorinostat, that's actually used in the clinic as an anti-cancer therapeutic, right? Because it inhibits deacetylase enzymes. And because we've got aberrant expression of these enzymes in cancer cells, it kills cancer cells specifically and not normal cells. Yeah.
And what does class 1 and class 4 mean here?
Yeah, so there's 4 classes of deacetylase enzymes: class 1, class 2, class 3, and class 4. Class 3 are known as the sirtuins. I don't know if you've heard about them or if your listeners have heard about them. The sirtuins are a different class. So class 1, 2, and 4 are metal dependent, right? So they depend on zinc to function. And they're deacetylase enzymes. The sirtuins are involved in longevity and other things, metabolism. Okay? Just it's a little bit confusing, but the 1, 2, and 3, what that means is that if you can inhibit these class 1 and class 4, right, you're inhibiting the majority of the enzymes. So a pan HDAC inhibitor, such as Vorinostat, means it will work to kill this cancer cell specifically. That's all. Yeah.
The other thing which I find interesting is that the combination of doxorubicin and Vorinostat showed enhanced DNA damage, specifically in K562 cells. And how might this translate to clinical applications?
Yeah, so, well, as I said, right? So doxorubicin is a frontline chemotherapeutic. So it's used for chemotherapy. And Vorinostat is also a drug that's used in the clinic, right? So you can combine the two. What happens is Vorinostat is a HDAC inhibitor, and as I said, it causes acetylation. Now, acetylation opens up the DNA. It decondenses the DNA, right? And that's why you get increased transcription. Now, if it opens up the DNA, there's more binding sites for doxorubicin to bind and do the killing. Because doxorubicin binds to DNA, right? So by opening the DNA with a deacetylase inhibitor, you get more doxorubicin in there, and there's more sites for the doxorubicin to do damage on the DNA because of the landscape of the DNA opening. Yeah.
Right. So these two drugs that we talked about right now, these are prescribed for any type of cancer or just CML?
So doxorubicin is widely used.
Mm-hmm.
Widely used. Vorinostat is not front line; it's a second line and third line. Okay, so if conventional chemotherapy doesn't work or if you become resistant, that's when they'll think about vorinostat. So it's not a front line cancer therapy; it's a down the line, right? It's if you've got no other choice, you might put in vorinostat. Yeah,
Right.
because
I
sorry.
was thinking like, if we have Gleevec and it works so good, like so perfectly, why do we want to like uh, do other drugs with CML patients? But I think this is like one of the areas that K562 can be applied to like any type of cancer, not just CML.
That's correct. That's correct.
Mm-hmm.
And also, as you said, with Gleevec, not a lot, but a proportion of patients will become resistant eventually, right? And they'll go into blast crisis, which is the end stage. And also, it is a management tool, right? So even though it works very well, beautifully, and I've seen people on it for 20 years and they're doing beautifully, even though it works very well, it doesn't cure the cancer. It just manages it, right? So the cancer, if you stop the drug or if you become resistant, then you've got no other choice again. Yeah.
Mm.
Mhm.
Right.
Mhm.
You also did a microarray analysis, which showed that cultured K562 cells have shifted away from patient-derived leukemic cells. Can you also talk about that?
Yeah, of course. And that's as you'd expect, right? So if you're getting chromosomal translocations and you're getting genetic drift, we saw that with at the single cell, at the not single cell, at the single gene, right? So when we did gene analysis, we saw that different genes express differentially. Okay, so that's all that means. That if you take a set of cancer patients, blood from them, as you talked about before, and you sequence it, the expression of genes will be different to the K562 cells. There will be some variability, right? And that's expected, as you'd expect. Yeah.
Right. And one last thing from paper, which I think is cool, is that you identified 5 genes involved in CML progenitor cell survival that are independent of the BCR ABL1 gene fusion that we talked about. Why is this finding potentially important for therapy?
So this is important because we know that the cancer stem cell hypothesis, where there is a progenitor cell that's then driving the cancer. So, and again, this comes back to the management question, right? Where you can manage it with Gleevec but not treat it. It's maybe that Gleevec's working on all the differentiated cancer cells, right, at a different phase to the one that's actually driving the cancer underneath, right? And you find that with other cancers as well, where there's a cancer stem cell which is resistant to therapy. However, you can kill the bulk tumor cells, right, that are differentiated, but then there's an underlying stem cell that you can't treat. And that's the issue there. That's where that might be important.
And so when you like look into the future, like moving forward, do you have any like um, idea that maybe like how K562 cell can be used in future research? Do you see any trend, like a specific use case that might be very popular soon in the future?
Yeah. Look, for cancer therapy, because of how easy they grow and how well characterized they are, despite these changes that I'm showing in my paper, right? They're very well characterized cell line and have been used for many years. They're a workhorse. They're going to be used for cancer research in general cancer, not just CML, for making targeted drug therapies and all sorts of things, right? They're going to be continued to be used. Um, and as I said, they were the workhorse for CAR T cell development as well. So there's a lot of uses for K562 cells, and the enormous numbers that you can generate in very short periods of time is a big advantage for cancer research. So yeah, yeah.
So any K562 cell that we have like right now in 2025 is, well, basically like they have been growing since 1970s, right? And
Yeah, so it might be a little bit more complicated, Mike, because what you might have is you might have labs that have frozen aliquots
oh, okay, yeah.
over time, right? So,
Right.
different
Yeah, okay,
well,
that clears up everything. Yeah.
yeah,
Because I was
well,
wondering, like, if we
Mm,
just want to keep using them, like at some point they're just
mm,
gonna be so different that like it
yeah,
doesn't even.
yeah. But you'd have different passage
Right.
numbers, so there'll be a lab somewhere that will tell you this is passage number 2 or passage number 10, right? So you'll have, yeah.
Right.
But you're right. What happened with HeLa cells, right? The big controversy with the HeLa cells eventually changed, and all this research, all this cancer research wasn't in with HeLa cells in the end, and it was, there was a big controversy. But again, that's a different topic. Yeah.
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 But I mean, like, even if it does happen, like based on what we talked about, I think it's not going to be difficult to establish a new cell line, given
Correct.
everything we know.
Yeah. You're right. Right. It wouldn't be very difficult. No.
Right.
Mhm.
And awesome. I think I've learned so much. And as we wrap here, after almost 3 decades of working with K562 cells, what advice do you have to researchers, like grad students like me, who are just beginning to play with these cells for the first time in their
Yeah.
research?
I think that if your supervisor tells you to use K562 cells, listen to him, like I did, because they are beautiful cells to grow, very easy to grow. And despite my paper and other papers have showed all this genetic drift and all this stuff, they are very, very well characterized. They divide every 20 hours, so you'll get a lot of cells for your experiment. You'll never run out of cells. You won't have an excuse, I couldn't do my experiment because I didn't have enough cells. There will always be enough cells. That's my main point. They're easy to grow, and they're actually a pleasure compared to some adherent cells or some normal cells which take 3 days to divide and need all this special media. Yeah, they're good cells.
Amazing. Dr. Karagiannis, thank you so much for this interview and all your wisdom on K562 cells. And let's just end this with this question. What advice do you have for me as a PhD student?
As a PhD, should you finish your PhD? No. So I'll tell you, Mike, 3 first author papers and a presentation, right? So you've got to keep that in mind, because to be honest, my son's just finished his PhD now as well. So, and I was telling him from the start, right? 3 papers and 1 presentation. That's what you need, right? So yourself, because I know I was young as well one time, don't forget. Yourself and other young people, you're very ambitious, and you have all these dreams and all these plans and all these ideas for experiments. Don't do experiments for the sake of doing experiments, right? Don't just do the experiment and say, and definitely, unless you're going to go on a paper. So if your lab is collaborating with another good lab and you're going to get a good paper, do that work, right? Because you'll get a very good paper, right? That's important. But don't just do work for the sake of doing work. I'll do this experiment, see what happens. I'll do this. Have a specific aim of why you're doing an experiment, and have a goal of publishing a paper as a first author, because that's what counts, right? In the end, you'll need 3 papers and a presentation, right? That's what you need to aim for. That's your goal. Of course, as I said, if your supervisor has good collaborations and there's an opportunity to get on a Nature paper, for example, you've got to go for that opportunity. Take the opportunity, right? Especially if you just have to do a simple experiment with K562 cells. But But that's my advice. My advice is be focused on the end goal. Otherwise, I've seen PhDs where it can drag out for years, like 6 years or 7 years and 8 years. And it's not a good thing. I mean, try to get it done. I think for candidates, 5 years. Is that correct? Or
Yeah.
5. Try to get it done within the 5 years. As a three first author publications, 1 final defensive thesis presentation. Keep it in mind. Don't deviate too much, right? I know you're excited and you want to do all these things, but you've got to hold yourself back and think, okay, I'm just excited. I'll do the experiment that's going to get me a paper and give me a story at the end, right? Yeah.
Right.
Yeah.
Amazing.